Many thanks for having me, and also a special thanks to Hannah, Julia and Alex for organizing such an interesting satellite symposium. I'm very happy to share some of our findings with you, but upfront I'd like to say that I'm a neuroscientist and not an evolutionary biologist, so please forgive me for not daring to interpret anything in terms of evolution. Chronological age is just a number. I'm sure you're familiar with this saying, and actually science tells us that there is a lot of truth in this because biological age most of the time does not correlate with chronological age, the factual age based on the individual's birth date. So people of the same chronological age, maybe here in the middle, can be very healthy and live as they were 20 years younger, much like this elderly couple down here, but they could also be on the upper side of this diagonal and suffer from a poorer health than the average couple of their chronological age. And today, the best marker we have for both chronological age and biological age is DNA methylation in humans. This epigenetic marker consists of the addition of a methyl group to the cytosine base of the DNA. And while cytosine methylation is part of normal base modifications that we can find in many species, it shows a specific pattern that is related to age. So this is also very often referred to as the epigenetic clock, since it correlates with chronological age and predicts lifespan most accurately in humans and in other greater apes too. But it is also noteworthy that the performance of distinct biomarkers, could also be others such as telomere length, differ from species to species because also the mechanisms underlying specific modifications can differ greatly. Now we know that a major reason for the discrepancy between biological and chronological age is that organisms are greatly influenced by environmental exposures such as smoking, drinking, or diet in general, also psychological stress, but on the contrary also positive or beneficial exposures such as a healthy lifestyle with a lot of sports. And they can then have a lasting impact on our organisms. We now know specific examples of such phenomena influencing the epigenetic methylation clock. For instance, space travel was shown to slow down the epigenetic aging as measured in blood cells recently. And the group of Elisabeth Binder in Munich demonstrated that stress, for instance in form of early life stress, can have the contrary effect and accelerate the epigenetic aging. It is now widely recognized that stress has lasting impacts on individuals, but by now we know that in addition it can also impact germ cells, and this can then have consequences on offspring metabolic health and behavior. So what if besides methylation exposures also affect other epigenetic markers? This seems very likely given that besides DNA methylation we can also find histone modifications and also non-coding RNA in mature sperm cells. We also know that environmental exposures not only impact DNA methylation but also other markers. Alterations in the transcriptome, in the proteome and in the metabolome have also been suggested as markers for biological age. I will present you with evidence that we collected of how experiences of different forms of stress affect epigenetic marks, but under the premise that the older one gets, the longer the list of impacts one is exposed to. But with a disclosure that we don't really study biomarkers per se. Instead, we go one step further from the level of mere markers, and I will show you how these markers are actually associated with phenotypes in the offspring. I was extremely lucky to have one of the few robust models of transgenerational inheritance at hand which is also highly relevant to humans. In brief, it involves 2 weeks of traumatic early life stress, and it is based on a well-established maternal separation paradigm. The male gets stressed in F1 and then never again in any other generation. Being a Petri-line model, the females are always unexposed. It excludes confounders such as altered maternal care as a transmitter. Our studies in this model led to a range of very interesting observations. We saw, for instance, that there are intergenerational alterations in sugar metabolism and serum small RNAs. We observed altered cognitive performance and increased behavioral flexibility across generations, and also changes in brain metabolites. The reason why I'm mentioning this model here explicitly is because it set the basis for the research since. In particular, one of my key findings that has moved the field from a more descriptive to a more mechanistic level. DNA methylation and histone post-translational modifications that have also been studied widely in this field do have substantial limitations when it comes to germline inheritance, because they get mostly erased during waves of epigenetic reprogramming between generations. We were the first to show that sperm RNAs are changed in response to early life stress, and I was also the first to causally prove the involvement of sperm RNAs or changed sperm RNAs in this transmission process. We achieved this by injecting sperm RNAs into naive, unexposed, fertilized oocytes. And then we could generate offspring that was then tested on the same tests as we would usually employ in natural MSU as offspring. Since the publication of our work, many other labs around the world have picked up on this idea, and this has resulted in a host of very interesting publications describing the impact of all different kinds of exposures such as chronic stress, also pesticides, stress hormones, fluoride, exercise, you name it, on small RNAs in sperm and their potential to transmit information to the offspring. I told you before that others have considered circulating metabolites as biomarkers, and we were wondering where the altered sperm RNAs in our MSUS model, this early life stress model, were coming from, because sperm is considered transcriptionally silent. So the changes that we observe cannot originate from within. Recent reports suggest that sperm is capable of acquiring RNA from its surrounding via epididymisomes. These structures are exosomal-like structures originating from the epididymis, the structure that the sperm is passing through during maturation. So we were asking whether MSU's sperm RNA originated from somewhere else in the body. And we started pursuing the hypothesis that early life stress affects RNA elsewhere in the body, then this would be circulated via serum and eventually being taken up by sperm. To investigate whether altered serum factors are functionally implicated in the transmission of the early life stress effects, we isolated serum from adult stressed males and from control males, and intravenously injected it into naive age-matched controls. We then bred these animals to obtain offspring, and this offspring could then again be tested on similar tests as have been employed in natural offspring of early life stressed males. You can see the effects in natural offspring of early life stressed males: a reduced body weight in adulthood and also a dampened glucose response during a restrained stress. But amazingly, the resulting offspring of the serum injections also showed a similar reduction in weight and altered glucose levels in response to a stress challenge, indicating an induction capacity of those serum factors for the transmission process. But when looking at the serum RNA, we couldn't detect any major changes, neither in small non-coding RNA nor in long non-coding RNA, using next generation sequencing. As mentioned before and also demonstrated in the MSUS model, stress can affect metabolism. And additionally, we know that stress is mediated by hormones, and besides hormones that are made up by peptides, there is also steroid hormones that are made up by lipids. And so to assess lipids in an unbiased fashion, we used the mass spectrometry approach for a metabolomic screen in plasma of these early life-stressed males. We observed expected changes in steroidogenesis in the directly exposed males, but the most striking changes were also observed in fatty acids and also bile acids. And these changes extended also to the next generation. We detected similar alterations in a cohort of children from an SOS children village in Pakistan that have experienced early life stress in form of paternal loss and maternal separation. Most interestingly, a common target of all these affected metabolites were PPAR receptors, nuclear receptors. To test whether serum indeed was functionally affecting PPAR receptors, we then turned towards GC1 cells. They are at the stage of becoming primary spermatocytes and are immortalized. And we transected them with a reporter plasmid for PPAR activity. After exposing them to serum of MSUs, early life stress males, we indeed saw that they show higher PPAR activation as read out by this plasmid reporter assay. We also observed that PPAR messenger RNA expression was elevated in MSUs sperm. PPAR protein levels were also increased in the epididymal white adipose tissue, and several PPAR targets showed altered expression in the liver, a primary site of PPAR action. So altogether, these findings point towards altered PPAR signaling in general. So following the same injection paradigm as we used for serum injections, we then injected a dual PPAR agonist tesaglitazar into males and molecularly analyzed the sperm. We saw that indeed, both for transposable elements, but also for other RNAs, coding RNAs, there were very similar changes induced in this sperm as we were able to see in MSU sperm. To test the possibility that metabolite-induced alterations can mediate transgenerational effects, in our model, we looked at the offspring of the males that had received such injections. And strikingly, these males here at the bottom display some of the metabolic features that we can also observe in a natural MSU's offspring, which, as a reminder, is displayed again at the top of this page. So a reduced body weight and altered glucose response in the glucose tolerance test. So overall, we conclude that early life stress can alter serum metabolites that affect PPAR signaling, and that this altered PPAR signaling is crucially involved in the transmission of the early life stress-induced phenotype. Now, changing gears here, we've heard before that stress can alter the epigenetic DNA methylation clock in humans. That study also had used dexamethasone, a stress hormone receptor agonist, to mimic those effects. And we also use dexamethasone to mimic stress, but we do not only collect sperm and serum for molecular analysis 14 days after injection, but we also use the sperm for in vitro fertilization with naive, unexposed oocytes to generate offspring. And importantly, in our model, we only use a single dexamethasone administration to activate the glucocorticoid receptor, the stress hormone receptor, and can then precisely coordinate the molecular analysis with the timing of the fertilization during IVF. So we know that the molecular changes we observe are those relevant at the time of fertilization of the egg. When we then phenotype the resulting offspring, we can observe that they show a range of metabolic alterations. For instance, here on the insulin tolerance test, which is again injecting a dose of insulin and looking at glucose levels over time, but also generally on the body mass index, which consists of the length and the weight of the animals. Now, when looking at the sperm markers with next generation sequencing, we found alterations in several classes of sperm RNAs, including microRNAs, tRNA derived fragments, and also broad changes in ribosomal RNA derived fragments. Some of the altered sperm RNAs were also altered in serum exosomes, suggesting that they are either altered systemically or that sperm changes are induced by uptake of serum exosomes. And strikingly, the changes of the single dexamethasone treatment were apparent as soon as 3 hours in sperm, however only in cauda epididymis sperm, the place where mature sperm is stored, not in the caput sperm. And this, as a side note for the sperm RNA enthusiasts, indicates that the sperm most likely is not required to pass through the epididymal tract in order to acquire this change. This is, according to some studies, the case for other tRNA derived fragments. And to add yet another interesting class of sperm RNAs to the long list of potential biomarkers, we also reported for the first time changes of circRNAs in response to our stress mimic. Here, circTASP1 and circDND1. CircRNAs are produced by alternative splicing, and they are particularly interesting since they are highly stable due to their circular nature, and could thus be protected during sperm to oocyte delivery and thereby then affect offspring embryonic phenotype. They functionally can either be translated into small peptides or act as microRNA sponges, thereby regulating targets of microRNAs. And in our model, we indeed found upregulation of SAT1. This is a diamine acetyltransferase, as assessed by single embryo sequencing at the 2-cell stage. SAT1 is a target of microRNA 31105P, a common circRNA target of both our altered circRNAs. And this indicates that potentially our upregulated circs are transmitted to embryos, sequester certain microRNAs, and then this in turn leads no longer to a downregulation of their targets, and then becomes apparent as this upregulation. So turning now from external to internal stress, we have also looked into a model from Julia Schroeder's group on the impact of paternal age on offspring fitness in the sparrow. This is an outbred setting, of course, very different from our inbred setting before in mice, in these inbred mouse lines. And the effect is observed in a wild population on Lundy Island. But for our experiments, we used captive sparrows from Julia's aviary at Silwood Park. And while we did not find differences in the sperm microRNA and tRNA fragment payload between prime-aged and old-aged sparrows, we do provide the first description of small RNAs in this unconventional model organism. We know that other classes of sperm RNAs can also be functional in transmitting phenotypes. So we hope that new annotations of the sparrow genome might facilitate analysis of other classes of sperm RNA in the future. So this could still lead to a potential explanatory mechanism for the transmission of the effects of age on offspring fitness. So I'd like to summarize and speculate a little bit. I've shown you that experiences can shape the germline epigenome and thereby also have an impact on offspring phenotype. And of course, the older we get, the more experiences are accumulated. So I'd like to pose the question whether our observations in sperm markers could have a value as a biomarker. And if adaptive or also maladaptive, could germline alterations that have phenotypic outcomes hold then an evolutionary impact. And with this, I'd like to thank you for listening. Also, a special thank to all the collaborators, upfront also to Julia Schroeder, and I'm happy to take your questions.